Let's get right into it. Welcome to a journey through the silent assassins. Plants you're lucky aren't big enough to eat you. But for some bugs and other creatures, their fate is not so similar. These secret predators wait for you to make one wrong move, and it's on to the next life for you. This botanical brigand preys not on the plantain, but on protein, drawing nourishment from the nitrogen-rich bodies of insects. Its menu is meticulously curated. Ants march to their doom, beetles blunder into botanical bear traps, and spiders find themselves ensnared in silken green graves. The flytrap favors the flyers and the crawlers, each a bite-sized banquet enriched with essential nutrients that are otherwise scarce in its soggy soil sanctuary. Behold the hinged harbingers of death, these leafy lobes. Each trap is a marvel of morphological magnificence, evolved expressly for entrapment. The leaves, dual-lobed and rimmed with spine-like cilia, snap shut in an instant upon the right provocation. The trigger? Delicate trichomes, sensitive as a poet's soul, awaiting the twice-told touch that signals the presence of prey. This mechanism is powered by rapid changes in turgor pressure within the cells, a shift so swift it seems the leaves leap at the chance to clasp their unwitting guests. The trap's closure is but the opening act in the drama of digestion. Upon the unsuspecting touch of an ant or the unsuspected trespass of a moth, the trap springs its surprise, snapping shut in less than a second, a morbid applause for the performance of the prey. But this is no immediate mastication. The trap meticulously seals itself shut, ensuring none of the digestive concert within is wasted. The process is punctuated by periods of stillness as the plant assesses the struggles of its captive, only secreting its digestive enzymes once sure of the catch, a parsimonious patron of its own energy. Within the closed confines of its cellular chamber, the Venus Flytrap conducts a chemical symphony. Enzymes like proteases, phosphatases, and chitinases are the maestros here, breaking down the bodily constituents of captured critters. Proteins are passed into peptides, chitin into simpler sugars. This digestive process is finely tuned, the flytrap extracting, absorbing, and assimilating nitrogen and other nutrients directly into its tissues, a botanical version of bloodletting that sustains and nourishes. In the nutrient-poor peat of its native habitat, the flytrap finds the feast of sunlight and soil lacking. It hungers for more: nitrogen for the amino acids that build proteins, phosphorus for ATP and DNA, potassium for its cellular functions. Each insect ingested is a packet of these precious materials, a nutrient nugget that supplements the Spartan fare of its sandy soils. This nutritional necromancy, turning prey into plant substance, is what allows this verdant villain to thrive amidst the dearth of its domain. Welcome to the shadowed realms of the boggy breathers, where the Cobra Lily, Darlingtonia californica, stands sentinel, a serpentine sovereign in a kingdom of muck and mystery. This pitcher plant, cloaked in a guise green and ghastly, sips from the cup of carnivory with an air of aristocracy that belies its brutal bounty. In the gastronomy of the Cobra Lily, there is an air of exclusivity, an Epicurean elite that dines delightfully on the doomed. This carnivorous connoisseur is not for the common fare. It hunts the humming life of insects, flies, ants, mosquitoes, and such small-winged wanderers. Each guest is invited unwittingly to a nectar-lined gala, only to find the floor beneath them a slippery slope of deceptive demise. Behold the botanical brilliance of the Cobra Lily's lascivious lures. Its leaves, sculpted into hollow, twisted pitchers, stand like urns awaiting ashes. The pitchers are adorned with a flared orifice, mimicking a snake's gaping maw, complete with a forked leaf resembling a fanged tongue. A translucent dome caps the pitcher, drawing the curious closer with the false promise of escape, where there is none. The walls within are slick with secreted slickness, a slide into digestive despair for any that dare to delve too deep. The Cobra Lily practices a dark dance, a choreography of cunning that beguiles before it betrays. Insects lured by the sweet scent of nectar meander into the mouth of the pitcher, entranced by light filtering through false exits. They are tricks of translucence, guiding the gullible to their gut. As they probe for a passage, they slip on the slick inner walls, tumbling into the depths of the plant's digestive dungeon. Within the confines of these natural necropoles, the Cobra Lily undertakes the unmaking of its victims. Here, enzymes do not rush forth as a flood. Rather, they rise slowly like a tide, breaking down the bodies of the beleaguered into their basal elements. Unlike its carnivorous kin, Darlingtonia lacks the common digestive enzymes. It relies instead on a retinue of bacteria to break down the banquet, a microbial menage a trois of decay and digestion. In the poor-soiled provinces of its Pacific Northwestern palace, the Cobra Lily reigns over realms where nutrients are but a miser's handful. It seeks nitrogen and phosphorus, those alchemical elements essential for its leafy luxuriance and floral flourishes. Each insect ingested is an infusion of these indispensable ingredients, helping to sustain and sculpt the sinuous splendor of its serpentine silhouette. Your next silent killer, the Butterwort, Pinguicula, a subtle seductress in the world of botanical brigands. Dressed not in the flashy garb of her carnivorous kin, but in a cloak of deceptive simplicity, this green gourmet partakes in the pestilent feast with a finesse that belies the fatal finish of her flypaper foliage. The Butterwort's menu is a minimalist manuscript inscribed with the names of small, unsuspecting arthropods, gnats, flies, and small beetles, those minute meanderers of the air. Each becomes a patron at her leafy lair, lured by the prospect of a sumptuous sheen, only to find themselves part of the repast. In her subtle dining hall, the Butterwort prefers the petit four of pests, each a bite-sized bounty in the banquet of survival. Peer closely at the Butterwort's unassuming leaves, flat, greasy, and glistening like dew-covered dawn. These leaves are living flypapers, coated in a slick of sticky mucilage that glistens with deceptive invitation. The gluey glamour of these leaves is not merely for show, it is a meticulously engineered trap, each surface a sticky stage where the dance of death is delicately played out. The act of capture in the Butterwort's world is not a dramatic snap or a sudden swallow, but a slow seduction. Insects, drawn by the gleam of the glandular droplets, alight upon the leaves only to find their feet fettered in a viscid vice. The more they struggle, the more ensnared they become, each movement a step further into the fatal embrace of the Butterwort's botanical bind. Once an insect is secured, the Butterwort's leaves exude additional digestive enzymes, bathing the beleaguered bug in a biochemical broth. These enzymes, proteases, esterases, and others, work their quiet chemistry, breaking down the body into absorbable aminos and mere molecules. This process is not the rapid ravage of ravenous relatives, but a gradual, genteel gastronomy that ensures no nutrient escapes its subtle synthesis. Thriving in thin soils often void of sufficient nitrogen, the Butterwort supplements its sustenance with the protein-packed parcels provided by its prey. This nutrient necromancy allows it to flourish where others flounder, crafting a life in locales laden with limestone or other mineral-rich but nutrient-poor substrates. Each insect ingested is an incremental increase in the essential elements: nitrogen for growth, phosphorus for energy transfer, and a suite of micronutrients that mingle to maintain the metabolic majesty of this modest matriarch. Silent but deadly in the mysterious expanse where the trumpet pitcher Sarracenia reigns, a stately sentinel in the sordid swamp, its towering tubes a testament to the tenacity of the trap. Arrayed like organ pipes in a grand cathedral of green, these majestic moors marshal the wind and whisper tales of deceit and digestion. The trumpet pitcher's menu is a macabre mosaic, a collection curated with the casual cruelty of a connoisseur. It dines predominantly on flying and crawling insects, each unwittingly winging their way to woe, flies, wasps, ants, and even the occasional moth or beetle. These creatures, drawn by the deceptive call of nectar or the beguiling brightness of the pitcher's colors, find themselves part of a more permanent collection, their essence distilled into a digestible draught. Behold the botanical brilliance of the trumpet pitcher's structure. Each leaf a long, slender chalice, its hollow inviting and its rim or peristome scalloped like the edge of a particularly precarious precipice. Coated with a slick, secreted substance, this rim plays the piper's tune, luring the loiterers to a lip from which there is no leaping back. Below this deceptive doorway, the inner walls glisten with a glistering glaze, ensuring that ascent is as arduous as scaling glass. The approach to demise in the trumpet pitcher's realm is a descent into darkness. Insects enticed by the nectar notes along the rim meander into the mouth of the pitcher, slipping suddenly on the slickened slope. Once inside, the descent deepens. The walls, waxen and wily, ward off any would-be escape, guiding guests ever downward into the digestive depths where a pool of enzymatic elixir awaits. Within the belly of the beast, a slow symphony of dissolution commences. The trumpet pitcher secretes a suite of enzymes, proteases to pass the proteins, lipases to liberate the lipids, and amylases to annihilate the carbohydrates. This liquid lament transforms the trapped into the nutrients necessary for the pitcher's persistence. Moreover, unlike some of its carnivorous cousins, the trumpet pitcher often hosts a hive of symbiotic bacteria that aid in the breakdown of the bodies, a microscopic menagerie that munches what mere molecules might miss. Inhabiting soils sparse in nitrogen and other vital nutrients, the trumpet pitcher plants its roots where few others flourish. Each insect ingested infuses into its system a surge of nitrogenous nourishment, a sip of phosphorus, a draught of potassium, elements essential for the plant's photosynthetic prowess and foliar development. This digestive alchemy allows the pitcher to unfurl its foliage further, extending its reach into the rarefied realms of the nutrient-needy niche it navigates. Journey into the supernatural world of the Pameridea Roridula, an enigmatic entity within the plant kingdom, crafts its curious coexistence. This peculiar plant, not quite a carnivore nor a mere spectator in the saga of survival, stands amidst its fellow floral fancies with a flair unique unto itself. Pameridea Roridula is not the typical diner one might imagine in the carnivorous cohort. It rather hosts a banquet where the main course is served by others. This intriguing insect, the Roridula gorgonias, thrives on the plant's sticky surfaces, catching prey yet unable to digest them directly due to lacking its own digestive enzymes. Instead, it relies on its resident insects, the Pameridea bugs, to consume the ensnared insects. These bugs process the prey and excrete nutrients, which the Roridula then absorbs, turning what is a meal for one into sustenance for two, a true testament to the ties that bind in the biological ballet. Roridula is a plant that masquerades as a murderer, its leaves coated in a thick, sticky resin that glitters like dew under the dawn light. Each leaf is a labyrinth of stickiness, a deceptive path where insects wander with waning hope. This sticky substance is not just any trap, it is the stage upon which the drama of dependency unfolds, attracting and capturing a variety of insects, which then become the unwitting participants in this symbiotic soiree. The insects, lured by the promise of a verdant vista, find themselves fettered by the fierce stickiness of the Roridula. Here, the Pameridea bugs play their part, pirouetting among the prisoners, dining on the doomed. This interaction is less about the plant itself devouring the prey and more about it benefiting from the subsequent actions of its tiny tenants, the bugs. The plant's role is to set the table upon which the Pameridea bugs feast, recycling what they consume into a form that the Roridula can absorb. In this complex chain of consumption, the Roridula does not digest its catch directly, but absorbs the nutrients released by the Pameridea bugs as they excrete. This indirect method of nutrient uptake is a fascinating twist in the tale of trophic transfer, showcasing a sophisticated system where the plant's survival hinges not on its ability to digest, but on its capacity to collaborate. Thriving in nutrient-poor soils, the Roridula has carved a niche where it garners the necessary nitrogen and other nutrients from the bodies of insects, albeit indirectly. Each bug-borne excretion is a drop in the bucket of its nutritional needs, supplementing its photosynthetic practices and ensuring its growth and reproduction in an environment where direct nutrient uptake might otherwise limit its livelihood. Enter the domain where the yellow pitcher plant, Sarracenia flava, unfurls its chartreuse chalices, standing sentinel in the saturated sanctuaries of the south. This botanical buccaneer, cloaked in hues of harrowing yellow, orchestrates an opera of obliteration beneath the sunlit symphony of swamps and savannas. The yellow pitcher plant hosts a harrowing haute cuisine, its menu meticulously curated with the winged whimsies of the wetlands. Ants, flies, wasps, beetles, and even the occasional unsuspecting moth find themselves the featured guests at a feast where they will soon become part of the fare. Each insect, lured by the plant's vibrant color and the deceptive promise of nectar, unwittingly spirals into a botanical belly where escape becomes a flight of fancy too far to fathom. Each of Sarracenia flava's pitchers is a masterclass in murderous design, sculpted like a slender goblet that beckons to the beastly and the beautiful alike. The structure of these pitchers is both simple and sinister, an elongated tube topped with a flared hood, which serves to prevent dilution of the digestive brew by rainwater while also bewitching bugs into believing in a benign shelter. The inner walls are slick with a skid-inducing secretion, ensuring that any entrant's first step is a slip into the digestive depths below. The choreography of consumption enacted by the yellow pitcher plant is a ballet of betrayal. Insects are first enticed by the sweet scent and vibrant visage of the pitchers. As they alight upon the lip, drawn deeper by nectar guides and visual cues, they find their footing fails. The waxy, slippery surface of the pitcher's interior offers no purchase, plummeting them into a pool of potent digestive enzymes waiting at the base. Within this verdant vat, a vile concoction of digestive enzymes goes to work, breaking down the bodies of the befuddled bugs. Proteases and other hydrolytic enzymes cleave proteins into peptides, lipases dissolve fats into fatty acids, and chitinases degrade the exoskeletons of the chitinous. This cocktail converts the complex into the consumable, siphoning off the sustenance sealed within the sinews of the snared. Inhabiting nutrient-poor, often acidic soils, the yellow pitcher plant supplements its solar sustenance with sinister supplements derived from its prey. Nitrogen, phosphorus, and a suite of micronutrients are imbibed from the insectile infusion, vital for the plant's vigor and the verdure of its verdant vessels. This intake is crucial, compensating for the environmental scarcity that surrounds it and ensuring that each pitcher's promise of peril is not just a ploy, but a pivotal piece of its survival strategy. Venture into the watery realms where the waterwheel plant, Aldrovanda vesiculosa, executes its aquatic artistry, a submerged sorcerer in the liquid labyrinth of lakes and slow streams. This botanical buccaneer, cousin to the terrestrial Venus Flytrap, pirouettes in the ponds, each snap trap a marvel of motion and metabolism. The waterwheel plant partakes in an underwater banquet where its menu is meticulously etched with the effervescent effigies of aquatic invertebrates, water fleas, mosquito larvae, and other tiny denizens of the deep unwittingly waltz into watery traps, their movements triggering a lethal ballet that ends with their encapsulation. Each creature, small yet significant, contributes to the culinary cornucopia that sustains this submerged sentinel. Behold, Aldrovanda vesiculosa's formidable feeding mechanism akin to the bear traps of the deep. Each of its numerous bilobed leaf traps floats freely, tethered like a flotilla of fateful jaws, ready to snap shut at the slightest provocation. These traps are equipped with trigger hairs, which, upon the slightest touch by an unsuspecting swimmer, catalyze a rapid closure mechanism that seals the prey within milliseconds. A marvel of evolutionary engineering refined by the relentless rigor of aquatic exigencies. The ballet beneath the surface is a swift and silent spectacle. As aquatic prey brushes against the sensitive trigger hairs, the trap responds with a speed unseen in most of the plant kingdom. This rapid response ensures that the waterwheel plant capitalizes on the fleeting nature of its watery world, where potential prey is plentiful but perpetually passing, ensuring maximum nutrient acquisition from the transient troves that traverse its traps. Within the sealed confines of its snap traps, the waterwheel plant commences a concise concert of chemical breakdown. Here, in these miniature chambers of churn and change, enzymes are secreted to dissolve the detained. Proteases pulverize proteins, and chitinases cleave through chitinous exoskeletons, turning prey into a potion of digestible delights that diffuse directly into the plant's tissues, nourishing it in a nutrient-poor aquatic arena. Rootless and adrift, the waterwheel plant siphons its sustenance solely from the succulent soup it extracts from ensnared entities. This carnivorous consumption compensates for the dearth of dissolved nutrients in its aquatic abode. Rich in nitrogen and essential minerals, each captured critter is a crucial cocktail of life-sustaining substances, allowing the plant to perform photosynthesis with enhanced efficacy and to burgeon beneath the surface. The shadowed slimy assassin groves where the moccasin plant Cypripedium holds court with a quiet elegance, its slipper-shaped blooms nodding sagely among the understory. Known also as the lady's slipper orchid, this botanical beauty weaves a web of wonder and mystique with its deceptive allure and delicate demeanor. Unlike its carnivorous cousins, the moccasin plant does not dine directly on the bodies of the deceived, but thrives through a more subtle symbiosis. This orchid engages in a complex relationship with mycorrhizal fungi that interlace its roots. These fungi, adept at decomposing organic material, provide essential nutrients such as nitrogen and phosphorus to the orchid, which in return supplies the fungi with carbohydrates produced via photosynthesis, a mutualistic meal shared in the silent substrate of the forest floor. The moccasin plant's most striking adaptation lies in its iconic bloom, each petal shaped like a slipper, enticing not to ensnare, but to enchant. The flowers are engineered to attract specific pollinators, often bees, which mistake the blossom for a promising source of nectar. Intricate colorations and alluring scents draw these pollinators into the floral slipper, where the plant's reproductive strategies unfold with finesse. Once lured into the lip of the slipper, the pollinator finds itself on a slippery slope. The inner surfaces of the flower are sleek and waxy, designed to guide the insect downward towards a narrow exit through which escape is possible only by brushing past the orchid's reproductive structures. This ensures effective pollination as the bee struggles to find its way out, unwittingly carrying pollen from one bloom to the next, a dance of deception that benefits both dancer and observant. Beneath the soil, the moccasin plant engages in a less visible but vital exchange. Its roots intertwine with mycorrhizal fungi in a profound partnership. These fungi break down complex organic materials in the soil, making nutrients available to the orchid in a form it can absorb. In return, the plant's roots exude sugars, products of photosynthesis, that fuel the fungi. This exchange is a delicate duet, each partner sustaining the other in a cycle of growth and giving. In the dimly lit underbrush of its woodland home, where nutrients are sparse and competition is fierce, the moccasin plant's reliance on fungal partners is not just advantageous, but essential. It draws from the fungi a steady stream of mineral nutrition, crucial for its growth and the spectacular development of its blooms. This reliance underscores the orchid's sophisticated adaptation to environments where direct nutrient uptake might otherwise limit its flourishing. Enter the sun-scorched meadows where the dewy pine, Drosophilum lusitanicum, unfurls its sticky stars against a backdrop of arid air and stony soil. This botanical anomaly, more commonly known as the Portuguese sundew or the dewy pine, defies the conventions of its carnivorous cousins by thriving in the dry, unlike the typical wetlands favored by others. The dewy pine's menu is a testament to opportunistic dining, specializing in the insects that navigate the harsh landscapes of its Mediterranean home. Its sticky leaves become fatal attractions for ants, flies, small beetles, and occasionally even the wayward bee. Each insect, lured by the promise of dew-like droplets in a desert of dust, finds a final feast turned fatal, unwittingly contributing to the sustenance of this deceptive diner. Unveil the marvel of the dewy pine's feeding mechanism. Its leaves, slender and elongated, are adorned with glands that exude a thick, sticky mucilage. This gluey gloss is the plant's primary tool for capture, glistening in the sunlight like a sprinkling of morning dew, a sight both beguiling and deadly. Unlike the snap traps or pitfall mechanisms of other carnivorous plants, the dewy pine's strategy is one of static allure, where the victim's own movements seal their doom. The dance of death on the dewy pine's leaves is a slow and sticky affair. As insects contact the mucilaginous coating, they are immediately ensnared by its tenacious grip. The more they struggle, the more entangled they become, each effort to escape only tightening the plant's hold. This method of ensnarement ensures that once an insect touches the dewy pine's leaves, there is seldom a retreat from the reaper's reach. Upon securing its prey, the dewy pine employs a complex suite of digestive enzymes that seep out from the same glands that produce the mucilage. These enzymes, proteases, phosphatases, and others, begin the breakdown of the insect's body, liquefying internal structures and allowing the plant to absorb the resultant nutrient-rich soup directly through its leaves. This process of external digestion is both efficient and essential, converting the captured prey into critical nutrients within the confines of its arid environment. Thriving in nutrient-poor, often acidic soils, the Dewy Pine's carnivorous lifestyle is not merely a trait, but a necessity. The nutrients most crucial to its survival, nitrogen, phosphorus, and potassium, are gleaned from the bodies of its prey. These elements, scarce in the surrounding soil, are vital for the plant's photosynthetic processes and overall growth. Each insect consumed is a vital infusion of these essential nutrients, helping the plant to flourish where few others can. Fly into the cool, dappled shade of the woodland realm where the moccasin flower, or cypripedium, often referred to as the lady's slipper orchid, holds its court with delicate dignity. This charming chieftain of the underbrush, swathed in vibrant hues and curvaceous contours, weaves a narrative of nuanced beauty and beguiling biology. The moccasin flower, unlike its carnivorous floral counterparts, does not dine on the demise of the insects it lures. Instead, it thrives through a more profound partnership with mycorrhizal fungi that intricately entwine its roots. This symbiotic relationship forms the cornerstone of its nutritional intake, with the fungi breaking down organic matter in the soil, thereby mobilizing nutrients such as nitrogen and phosphorus, which are crucial for the orchid's growth. The moccasin flower's most remarkable adaptation lies in its bloom, shaped like a slipper, an evolutionary masterpiece designed to attract and momentarily trap pollinators, particularly bees. The vibrant colors and alluring scent draw these creatures in search of nectar. Once inside the slipper-shaped bloom, pollinators find themselves temporarily trapped, ensuring they come into contact with the plant's reproductive structures and facilitating effective pollination. The dance within the moccasin flower's blossom is a delicate ballet of enticement and escape. Pollinators, enticed by the flower's visual and olfactory cues, enter the slipper only to find that exiting is not as straightforward. The flower's design compels the visitor to wriggle and rub against the orchid's reproductive organs, thereby picking up or depositing pollen. This ensures cross-pollination, which is vital for the genetic diversity and survival of the species. Below the soil, the moccasin flower engages in a less visible but vital exchange through its roots, which are entwined with mycorrhizal fungi. This partnership is not merely parasitic, but mutualistic, where both parties benefit. The fungi facilitate the breakdown of complex organic materials, making essential nutrients available to the orchid. In return, the orchid supplies the fungi with carbohydrates derived from photosynthesis, sustaining their growth and perpetuating the symbiotic cycle. Inhabiting rich, often acidic soils of the forest floor, where direct nutrient competition with larger flora might otherwise overshadow a less equipped plant, the moccasin flower's reliance on fungal partners is a sophisticated strategy. It draws from the fungi a steady supply of minerals and nutrients critical for its growth, flowering, and overall health. This indirect method of nutrient uptake allows the moccasin flower to thrive in environments where nutrient availability is patchy or locked within complex organic compounds.